وحاليا الآن يسعدنا أن نستمع إلى عرض بسيط للبحث الفائز بالجائزة. تفضلوا.
أنا مش عارف أبدأ منين، بس أنا في قمة سعادتي النهارده بالتواجد معاكم، وشرف كبير ليا إن أنا أستلم الجائزة اللي أنا فرحان جدا جدا إن أنا استلمتها. أنا في قمة السعادة إن أنا أكون موجود وسط كوكبة من علماء النهارده، ويسعدني إن أنا أيضا أقدم لكم النهارده البحث اللي أنا اشتغلت عليه واللي بفضل الله نال الجائزة دي. For introducing my research, please allow me to transition to English. My name is Ahmed Badway. I'm a research assistant in the Center of Excellence for Stem Cells and Regenerative Medicine in the University of Science and Technology. And today I'll be presenting the research which won the Professor Dr. Hany Elsayed Marei Award for Excellence in Stem Cell Research. So as a kind of introduction, although this audience does not need any introduction for stem cells, because we were very lucky to listen to a wonderful introduction about stem cells from Dr. Hany, but as a brief introduction about stem cells come in different flavors, starting from totipotent stem cells, pluripotent stem cells, and multipotent stem cells. And these different flavors of stem cells play different roles throughout development. And the last type here, the multipotent stem cells, are the type of stem cells which gain much interest in the clinic, because as Dr. Hany mentioned, there is no ethical concerns when using multipotent stem cells. And these multipotent stem cells can be obtained from different sources, such as the lung, the bone marrow, the umbilical cord, as Dr. Hany also mentioned. And interestingly, the different type of stem cells, the different type of multipotent stem cells, also referred to as adult stem cells, have different therapeutic potentials. And in a publication we made in 2016, when we made a meta-analysis, when we looked at the 22 clinical trials that were published at that time, when we were asking, are there differences or which type of clinical stem cell therapy achieved the best outcome? In this meta-analysis, we found that hematopoietic stem cells achieved the best outcome, achieved the best therapeutic potential in type 1 diabetes mellitus, and umbilical cord blood achieved the worst outcome in curing or treating diabetes. And also, multipotent stem cells can be obtained from different sources, and multipotent stem cells, the most common type of multipotent stem cells are the mesenchymal stem cells and the hematopoietic stem cell. And the mesenchymal stem cell itself can be obtained from different sources, and the most common sources for obtaining mesenchymal stem cells are the adipose tissue and the bone marrow. And they are then being called adipose mesenchymal stem cells and bone marrow mesenchymal stem cells. So at that point, we initiated a study comparing the same type of stem cell, which is the mesenchymal stem cell, but obtained from different sources, if there are differences in their regenerative potential. And in another publication we published also in 2016, when we compared the therapeutic potential or the regenerative capacities between mesenchymal stem cell, the same stem cell, but obtained from different sources. And in this study, we found that adipose stem cells have a higher regenerative capacity than bone marrow mesenchymal stem cells, which was very interesting to us, as the same type of mesenchymal stem cell obtained from different sources are equipped with different therapeutic potential. And we performed the study in vivo in an animal model of hand limb ischemia, as well as many in vitro experiments as well. But the stem cell type which I gained much interest in is called the cancer stem cell. So I was very interested, and I transitioned into focusing on cancer stem cells, as currently people believe that cancer stem cells are the heart of the cancer and are the main cause of cancer progression and development. So cancer stem cells, there are different theories about the origin of the cancer stem cell. Where did the cancer stem cell come from? So there are different theories, and one theory is that the cancer stem cell arises from the local tissue rather than stem cells. So when the lung, for example, or the brain or any organ, all organs have their own local stem cells. And when the local stem cells acquire some mutations, they acquire cancer stem cell properties. And another story, which is also very well backed up is the theory that the cancer stem cells arises from a differentiated cell, from a terminally differentiated cell, but a terminally differentiated cell can acquire some mutations and it can de-differentiate and acquire some stem cell properties, and that's being seen also as a source of the cancer stem cells. So the cancer stem cells, why do we care? So the cancer stem cells are being seen as the main cause of metastasis also as well. And in many studies, metastasis is the leading cause of cancer-related deaths. In almost every cancer type, when patients are diagnosed with distant metastasis, the 5-year survival rate dramatically decreases, and cancer stem cells are being seen as the main reason of metastasis. And cancer stem cells, as you can see from this table, the markers for identifying the cancer stem cells are very diverse and are very dependent on the source of the cancer. So every different type of tumor has its own markers for identifying the cancer stem cell population. And this is clear from this table that there is no universal marker for cancer stem cells. And we were interested in breast cancer stem cells. And the breast cancer stem cell population is being identified by the CD44 positive and CD24 negative. So this was the seminal work published in 2003 by Al-Hajj and et al. So in this paper, this was the first paper which identified the cancer stem cell population. And in this paper, they find that as few as 100 CD44 positive and 24 negative cells were able to initiate a tumor when implanted in immune deficient mice. But as much as 1 million of the CD24 positive cells were unable to initiate a tumor in immune deficient mice. So this led to researchers identifying what's known as the cancer stem cell population, in which a certain population only within a tumor can initiate a tumor. So at this paper, the CD24 negative cells, only 100 CD24 negative cells were able to initiate a tumor in immune deficient mice. And these cancer stem cells are being seen as the cells which are able to undergo all the steps of the metastasis invasion cascade. The metastasis invasion cascade consists of many different steps, from extravasation, intravasation, and seeding metastasis in secondary organs. And the cancer stem cell population is being seen as the population which is able to undergo all the steps of the metastasis invasion cascade. So it's a very important cell within the developing tumor, and as I mentioned earlier, that these cancer stem cells, when they're responsible for metastasis, and this metastasis is the leading cause of cancer-related deaths. I was interested more in the cancer stem cell population, and I was very interested in studying the molecular regulators of the cancer stem cell state and what regulates these cancer stem cells. So I was very interested in the human telomerase reverse transcriptase. So the human telomerase reverse transcriptase, or the H-TERT, is the main subunit of the telomerase enzyme. And the TERT, or the telomerase enzyme, its function is by adding the TT, AGGG repeats at the end of chromosomes, which are referred to as the telomeres. And these telomeres play important functions in cell survival. And it's very important for cells to maintain their telomeres. And in normal tissue, upon every cell division, the telomeres are shortened until at some point the telomeres become too short that the cells are unable to proliferate again. But cancer cells, or at least 90% of cancer cells, acquire some mutation in the telomerase enzyme, and the telomerase enzyme becomes hyperactive. When the telomerase enzyme is hyperactive, it's always maintaining or elongating the telomeres, which maintains these cells in an immortal state, so the cells can keep proliferating without dying at all. And this is believed to be an important hallmark of cancer. So a very interesting phenomenon when I was deeping into the literature is that the cancer stem cell population, they have short telomeres, and this has been shown in different cancer stem cell populations. And they have short telomeres, although they express high levels of the telomerase enzyme, which is very interesting, and which led me to hypothesize that that the TERT, and the telomerase enzyme in the cancer stem cells has different roles beyond tumor beyond telomere elongation and maintenance. So for that purpose, first of all, we started by isolating the CD44 positive, 24 negative cancer stem cell population from the MDA MD231 cell line, and this was a FACS sorting experiment, and these cells were cultured in serum-free conditions to maintain their 24 negative state. And we started by, after isolating the cancer stem cell population, by first of all overexpressing the HTERT protein, and on the other hand down-regulating the HTERT protein as well. So the figure here on the left is a Western blot, first of all confirming that HTERT was successfully overexpressed in one population, and another population HTERT was down-regulated. And also we have qPCR data and immunofluorescence data which backs up and confirms that HTERT was successfully down-regulated and overexpressed on the other hand. So a very interesting phenomenon we saw when we down-regulated the HTERT is that these cells had a very dramatic change in their morphology, and they transitioned from the elongated mesenchymal phenotype into a round and epithelial phenotype, morphology I mean. So we saw that when we knocked down HTERT, the cells transitioned to an epithelial morphology instead of having their long mesenchymal shape. So this led us to hypothesize that HTERT might be playing an important role in the cancer stem cell population in what's known as the epithelial to mesenchymal transition. So the epithelial to mesenchymal transition, or the EMT, is a very important phenomena, and many work from the lab of Robert Weinberg, which I'm currently a part of, they have been showing that the cells, or the cancerous cells which acquire invasive properties or metastasis properties, they acquire a mesenchymal phenotype and they transition from an epithelial state to a mesenchymal state. So it's very well believed that the mesenchymal cells within the tumor are the cells which are responsible for metastasis, for drug resistance, and many other cancerous properties. And the epithelial phenotype is the more steady state. So the epithelial phenotype is believed to be the steady state, and the mesenchymal phenotype is believed to be the aggressive state within a cancer. And there are different markers which can differentiate between the epithelial and the mesenchymal cells. So epithelial cells, they express E-cadherin, a very known marker for the epithelial phenotype, and when they transition to a mesenchymal state, they acquire expression of markers such as N-cadherin and snail and slug. So when we saw that when we knocked down HTERT, we saw a transition to an epithelial phenotype, we started to deep into this more. And then we here see that the scramble cells here are the control cells. So the cancer stem cells in general, they are very mesenchymal because they are cells responsible for drug resistance and very aggressive. So the cancer stem cells initially are very mesenchymal. They express N-cadherin and snail and slug, but do not express E-cadherin. And when we knocked down HTERT, we saw that these cells lost their N-cadherin and snail and slug expression, and on the other hand, they gained E-cadherin expression. And the high cells, which the overexpressed HTERT, they even acquired a more mesenchymal phenotype. It's hard to see by the immunofluorescence, but the qPCR and the flow cytometry data show that the high cells, and I'll be referring to the high and low as high overexpressed and low as downregulated. So the high cells, they are even more mesenchymal. And then we started to ask, what happens if we get the mesenchymal cells and make them undergo an MET, which is the reverse of EMT, so the mesenchymal to epithelial transition. So when we had the high cells, as shown earlier, they're very mesenchymal, express N-cadherin, and don't express E-cadherin. But when we added a molecule called PD173074, which is a known molecule to induce the reverse of it, to induce an epithelial state. So when we added that molecule, we saw that the molecule works, and these cells gained E-cadherin expression and lost their N-cadherin expression. And this acquiring of the E-cadherin, acquiring of the epithelial state, made these cells lose their HTERT expression. And on the other hand, when we got the low cells, which were very epithelial to start with, they have high levels of E-cadherin and don't have the HTERT. But when we added TGF-beta, which is a known inducer of the mesenchymal state, that these cells successfully acquire the mesenchymal state first of all by gaining N-cadherin expression, and this gain in the mesenchymal state made these cells also gain some HTERT expression. which led us to believe that the H30 expression in cancer stem cells is mutually exclusive to the mesenchymal phenotype. And this figure, when cells undergo an EMT, they gain H30 expression, and when they undergo the reverse, the MET, they lose their H30 expression. So this led us to hypothesize that the H30 is very important for the cancer stem cell state, and for that we did some more characterization of important markers for cancer stem cells. And one important marker for cancer stem cells is ALDH1A1, which is a marker for cancer stem cells. And we saw that the low cells, they lost their expression of ALDH1, which means that they lost their cancer stem cell state. Another important marker for the cancer stem cell state is the beta-catenin, which is part of the Wnt beta-catenin pathway. And we can see over here that in the low cells, the beta-catenin is diffused throughout the cytoplasm. But when we look at beta-catenin pattern in the high cells, we see that the beta-catenin is localized into the nucleus. And beta-catenin localization into the nucleus is believed to be as a sign of activation. So when beta-catenin becomes active, it localizes to the nucleus. So this figure also shows us that H30 is very important for different cancer stem cell regulators, such as ALDH1A1 and Wnt beta-catenin pathway. And another property of cancer stem cells is what's known as the site population properties. So this is an assay which tests for the capacity of the cells to refuse a dye in this case, because cancer stem cells are known to have some pumps on their surface, and this is how these cells acquire chemoresistance. Because when the chemotherapeutic drugs enter these cells have some special property that they have pumps on their surface, and they can reject back the chemotherapeutic drugs which have entered into the cell. To do this, the analogy of this, we added a dye known as the Hodges dye, the Hodges blue dye, and then we tested the capacity of these cells to refuse the dye. And this dye is fluorescent. So we can see here also that the low cells have a decreased percentage of the site population properties, while the high cells, they even gain more site population properties. And to do the real experiment of testing the capacity of these cells to acquire chemoresistance, we added increasing doses of cisplatin and doxorubicin, very common chemotherapeutic drugs used in the clinic. And we can see that the high cells, they acquired more chemoresistance to increasing doses of these chemotherapeutic drugs, and the low cells acquired more sensitivity to chemotherapeutic drugs, which means also that the H30 molecule can regulate the chemoresistant properties of these cancer stem cells. And also to further characterize this more and more, we did a simple scratch wound healing assay, which measures the migration capacity of these cells, and also on the same trend, the high cells, they had more migration properties. And also another hallmark of cancer stem cells is their capacity to form tumor spheres when cultured in suspension conditions. We also did this experiment as well, and we saw that the high cells also, they acquire more capacity to form tumor spheres, more in number and more in diameter as well. And also when we're doing a simple colony formation assay, we can also see that the high cells acquire more colony formation capacities. And cancer stem cells are also very attractive in the clinic because these cells are very resistant to different insults. When we added oxidative stress and serum starvation to these cancer stem cells and just measured viability as a sign these cells are still strong and still being able to resist different insults, and on the same trend we can see that the high cells have also higher resistance to oxidative stress and serum starvation. And also, cancer stem cells have been shown in the literature to be able to differentiate in vivo into endothelial cells, which led many researchers to believe that these cancer stem cells are responsible for the angiogenic potential of the tumor. And we also see the same trend that by doing a tube formation angiogenic assay, we can see that the high cells the H-third high cells have higher tube formation properties compared to the low cells. And just to, my final slide is just to check the clinical relevance of the H-third and the EMT in clinical cases. So for that purpose, we accessed data from publicly available data in the TCGA, and this we looked at normal patients and cancerous patients, and we did see that in the cancerous patients they have more expression levels of H-third, and in the same hand the cancerous patients have lower levels of the E-cadherin, which is the CDH1, and they have higher levels of mesenchymal markers. So in clinical cases also, by looking at publicly available data, we can see that there's a correlation between H-third expression and the mesenchymal phenotype. So the conclusion is that in this research we've been working on, is we reported a critical war for H-third and cancer stem cells, and we showed that there is a reciprocal bidirectional coordination between the H-third and the mesenchymal phenotype, which is responsible for many of the cancer stem cell states. Properties. And then we also show that H-third is mutually exclusive to the mesenchymal phenotype. And when cells acquire mesenchymal phenotype, they acquire H-third expression. And similarly, when they lose the mesenchymal phenotype, they lose H-third expression. And the significance of this work, we believe, this work can be believed important because in clinics nowadays, there has been started some clinical trials to test the capacity of many telomerase-based therapies. Because of the importance of the telomerase enzyme in the H-third, there are many drugs being developed nowadays to target the telomerase enzyme. So we believe that the mode of action of these telomerase-based therapies can be achieved by when they target the cancer stem cells. These cancer stem cells transition from the aggressive mesenchymal state to the steady epithelial state, which can explain the success of some of the telomerase-based therapies. And this is the publication which we published in 2018 in Journal of Biology Open. And you can see here that this work is not a sole person work. I have been working with a very wonderful team to achieve this work. And this will bring me to the acknowledgement. And I would like to thank my professor and supervisor at Zawil City, Dr. Nagwa Al-Badri. She's the director of the Center of Excellence for Stem Cells, and she has been providing tremendous support for this project and tremendous guidance throughout the research course of this paper. And also I'd like to thank all my colleagues who have been also co-authors on the publication which I just presented. And I would also like to thank everyone else in the Center of Excellence for Stem Cells. And also, I see that being awarded the Dr. Hany Elsayed Marei Award for Excellence in Stem Cell Research is not only a great honor and privilege for me, but also it's an obligation for continuing to do hard work. Because receiving such a prestigious award in stem cell research while being in Egypt is not only just an award, but it provides hope for me and for many other young scientists working in the field of stem cell. And being recognized for the research we do in Egypt, it just makes us work harder and harder again to continue our excellence and to continue pursuing excellent research in the field of stem cell research. And we hope that one day, by our research and by these wonderful awards, that I'm sure also in the future many other wonderful scientists and researchers will also win this prestigious award. By these awards we can make a difference in the field of stem cell research, and if at least push the field of stem cell research a step forward and compete worldwide with our research. And with that, I'd like to thank you all for your attention and for having me here. It's been a great honor to be presenting my research in this wonderful audience. And thank you very much, and I'll be happy to take any questions if you have.
بالدكتور احمد الحقيقه يعني يكون يعني علماء المستقبل في مصر يعني كان يحافظ على هذه الشكل فخر ان انا انا لاحظت ان في البعض من حضراتكم دون حاجات يمكن عايز يعقب او حاجه في حضور، فانا ما هدعو الدكتور هاني نقل له اداره الجلسه في التعقيب وتلقي التعقيبات من حضرتك. اتفضل.
طبعا انا بتوجه بالشكر للدكتور احمد، وواضح التميز والبحث اللي بيعمله ده ببساطه شديده جدا بيدور على علاج جديد للكانسر، بثبت ان الكانسر ستيم سيل هي المسؤوله عن الانيشيشن او نمو الكانسر. فهو لا وجد ان احد الكانسر ستيم سيل الخصائص بتاعتها انها تتحول من ايبيثيليال لميزنكيمال او ميزنكيمال ستيم سيل، ودي اللي هي اجريسيف سيل وتبدا تعمل ميتاستاز. هو بيعمل ايه؟ هو عايز يمنع الستيج دي، وبالتالي وجد علاج للكانسر. وده اهميه ابحاث الخلايا الجذعيه، ان احنا مش بندور على علاج. ابحاث الخلايا الجذعيه تشابهت جدا، واصبح الكانسر ستيم سيل احد المكونات الهامه جدا لابحاث الخلايا الجذعيه. ودلوقتي احنا داخلين احدث حاجه في الاميون ثيرابي او الكارتي سيل، ده بقى هيعمل ثوره وعمل فعلا في مجال علاج الكانسر. بشكرك جدا يا دكتور احمد، وانا اتنبأ ونفتكر هذا اليوم دكتور احمد ان شاء الله يكون احد الحاصلين على جائزه نوبل باذن الله في الطب، انه واضح ما شاء الله وكمل. وفعلا الجائزه برضه بتفخر ان هذا المستوى من الابحاث ان احنا فعلا نجحنا او اللجنه نجحت في السلكشن الصحيح. ولو قبل ما اتوجه للغزال، لو في اي حد حضراتكم عنده استفسار للدكتور احمد او اي سؤال احنا تحت امركم. طيب احنا فاضل لنا حاجتين هنسلم اتفضل.
بسم الله الرحمن الرحيم. والصلاه والسلام على اشرف الخلق سيدنا محمد وعلى اله وصحبه وسلم. اولا انا في البدايه بشكر الدكتور هاني مرعي على هذا الجمع، واعتذر ان اتاخرت شويه بس والله يعني ظروف قهريه. بشكره وربنا يجعله في ميزان حسناته تجميعته هذه ودعوته دائما للعلم. انا ليا استفسارات بس بسيطه جدا. يعني انا اشتغلت حاجات على الستيم سيل ولو انها بعيد، لان انا شايف ان ما جيتش من اول المحاضره. موديلز كتير سواء كان اوستيوارثرايتس، سواء كان الزهايمر او الباركنسونيزم او الاوبستي، يعني هاو يو كان تريت باي ستيم سيلز. انا كنت بس هسال سعادتك، انا ماليش قوي في الدياجنوستيك ايدز اللي حضرتك استخدمتها عشان تعمل ديمونستريشن او ازاي ان الكانسر سيل بتتحول الى بسيلة سيل زي ما الدكتور مرعي عقب. انا بقول بس حضرتك هل في ديفرنت دياجنوستيك مينز او بعض النانو بارتكل ممكن تستخدم في هذا عشان تعمل ديمونستريشن؟ وهل يعني سكسس ان هي انت منعت او اللي شفت ان الخلايا دي ما بتنقسمش من ميزنكيمال سيل او اللي هي اللي هتبقى مور اجريسيف او الكانسر سيل؟ دي اول نقطه. برضه بقول لحضرتك يعني عمليه الدياجنوزس او ان انت بتقول ان فعلا ده هيبقى علاج كتير زي ما الدكتور هاني مرعي، انا شايف ان هو هيوز اذر مينز اوف اسسمنت، يعني ببعض الحاجات زي النانو بارتكل زي الفيروسكسيد نانو بارتكل محمل، او ان نستخدم بعض عشان اعمل ديمونستريشن فعلا للافكت اهو. بعض الدياجنوستيك ايمج اللي زي الام ار اي اساسا مع محمل من بعض حاجات النانو بارتكل او نيوكلير سينتوجرافي، كل دي ايدز تساعدنا اكتر ان انا اقدر اعمل ديمونستريشن واقول ان انا فعلا الخلايا دي، مع ان انتم جايز يكون شغل الهيستولوجي او البايو تكنولوجي وهيبقى ديمونستريشن اكتر، انما انا بتكلم من وجهه الجراحيه اليوزز اوف بعض الدايجنوستيك ايدز او بعض النانو بارتكلز المحمله ان هي تديني ديفينيت دايجنوزس وبروجنوزس لساتش كيس. وشكرا جزيلا على لقائك، ومره ثانيه بشكر الدكتور هاني مرعي وشكرا جزيلا. السلام عليكم.
يعني انا ممكن اعقب بتعقيب بسيط، اللي هو فعلا السؤال بتاع حضرتك والتعقيب بتاع حضرتك في غايه الاهميه، وفعلا نقطه جميله جدا ان احنا نشير ليها، وده اللي احنا فعلا شغالين عليه دلوقتي، ان احنا بنحاول نجرب الريسيرش بتاعنا في انيمال مودلز، ان احنا بنحاول نجيب telomerase based therapies في انيمال مودلز، ونشوف اذا كان the telomerase based therapies can in vivo also like recapitulate what we see in vitro as well. So this is a very important step which we're doing right now already. And also, I'm not that familiar with nano-based therapies, but with this study which we worked on, we were aiming at understanding how cancer stem cells are being regulated. So, and I believe that this is the first step of treating cancer, because there are many steps of cancer progression which we don't know about, which we're unfamiliar with. So I believe that by understanding the cancer stem cells, we would be able to treat them in the future. So this is what was the aim of our research, was to understand the cancer stem cell state and what are the important or the critical regulators of cancer stem cell. And this is what we worked on. We were successfully able to find a molecule which is very important, which is the H-TERT. And our future endeavors, our future experiments will be aiming at testing this as well as in vivo as well. And thank you very much for your time.
قبل ما نتوجه للغداء بس فاضل الشيك، وبعد كده هستاذن حضراتكم نصور صوره جماعيه وبعد كده نتوجه. طب ممثل الدفعه اللي انا اتخرجت منها سنه 85، حبايبي طبعا بتوع طب بيطري اسكندريه بيشرفوني وجايين من اسكندريه، فدكتور احمد طبعا كان الكابتن بتاع الفريق فعايز يقول كلمه فما اقدرش طبعا.
فخر لنا ان يكون هذا العالم القدير من جامعه الاسكندريه وخاصه كليه الطب البيطري الاسكندريه فينا ان شاء الله. انا نفسي في العلم ماليش في العلم عشان احقق للحق، لكن هذا الانسان الذي امامكم لم يتفوق بحرف خاطئ على مدار ال 35 سنه اللي عشرت فيهم صداقه و4 سنوات، لانه كنت حضرتك للاسف متقدم عليه بسنه يعني في اعدادي، مش متفوق في العلم طبعا عشان ما حدش يفهم. بشكر الاستاذ الدكتور هاني السيد مرعي على هذا التقدم الذي يجعلنا ان نفخر به على مستوى العالم وليس على مستوى مصر. ونشكر الدكتور احمد الفائز الذي يعد من اكاديميه كبيره جدا وهي العالم المرحوم رحمه الله عليه زويل، وان شاء الله هنشوف زويل وهنشوف الباز. وانا بتوقع باذن الله كما وعد الدكتور هاني مرعي ان هذه الخلايا الجذعيه ستقوم لها اثر فيما بعد. الكوليرا من 50 سنه شوف كان تاثيرها وكان بيموت فيها مئات الالوف، اصبحت ما شاء الله بفضل علمائنا وعلماء العالم لها الذكر. فكنا ان احنا نتفوق في علوم الخلايا الجذعيه، فان شاء الله ده فخر لكل مصري بهؤلاء العلماء. انا سعيد جدا في هذه الصحبه العلميه الجميله، وخاصه عميد الكليه المحترم والساده الافاضل. ونفسنا كده زي ما سمعنا عن الدكتور الاستاذ الدكتور هاني مرعي هذا التفوق العلمي وهذا النشر الجميل الذي نفخر به المصريين على مستوى العالم، نسمع بردك تطورات بقيه معلش مع احترامي الشديد لجامعه منصور على دماغي من فوق، التفوق العلمي لجامعه اسكندريه من الدكتور الجوهري والدكتور كمال ابو العز. وكل سنه وانتم طيبين كامل، وكل سنه وانتم طيبين وشكرا. ولنا
بشكر الدكتور احمد جاب الله كابتن الدفعه بتاع الدكتور مع تسليم الجائزه الماليه مقدمه ولتحمل اسم الدكتور هاني السيد مرعي، قدم منه قيمه 50,000 جنيه مصري الى الباحث الدكتور احمد البدوي الباحث باكاديميه زويل للعلوم والطب. تهانينا للدكتور احمد مرات ومرات. موفق دائما الدكتور احمد ان شاء الله.
فرحة 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 هي 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 ووو